Good afternoon to all the EAPCET aspirants. So yes, today we are going to deal with the unit microbiology, wherein we are going to start with bacteria. Okay. So here if you observe the introduction of bacteria, first microbiology is the word which is derived from Greek, wherein micros means small and bios means life, logos which is steady. Okay, it deals with scientific study of microorganisms, that is microbes. This also includes protozoa, microscopic algae and fungi. Okay, like yeasts and molds, unicellular fungi and multicellular fungi. Okay, microbes are omnipresent. They are ubiquitous or cosmopolitan in distribution. Ubiquitous or cosmopolitan in distribution. Okay. Historical contribution, if you observe, Anton van Leeuwenhoek from 1632 to 1723, he has done early works on microorganisms, and it is Louis Pasteur from 1822 to 1893. He has developed pasteurization and vaccine techniques. Robert Koch from period is from 1843 to 1910. He has contributed foundational work on microbiology. This is a brief intro on the discoveries of microbes. Who has discovered the first living cell? It is Anton van Leeuwenhoek. Here we go with discoveries. Anton van Leeuwenhoek is a Dutch merchant. He has built some small hand-held microscopes. These are observed through lenses, and he has discovered a world of invisible creatures and named them as animalcules. Okay, he has named them as animalcules. Microbes were named as animalcules. They are found. He has found these in water droplets, particles of soil, teeth scraping in the year 1674, and he has sent his observations and drawings to Royal Society of London. These were the first visual representations of bacteria and protozoa, and that is the reason he is regarded as father of microbiology. His most powerful microscope has a magnificent of 266x. The microscope which he has given, it has a magnification of 266x. Here we go with intro of bacteria. Till now we have already learnt, and it is the scientist who is Louis Pasteur. He has showed bacteria as chemical factories. These are the chemical factories that can change in nature, that cause change in nature, and he is known as father of bacteriology. Robert Koch, he has linked the bacteria to infectious diseases via germ theory of diseases. Here we go with bacteriology. What is bacteriology? Study and research on bacteria is addressed as bacteriology, and this will be its ecological distribution. Bacteria can survive in any extreme environments, even in Arctic snows. They are present in any extreme environments like Arctic snow, volcanic ash and hot water, and even in sulfur springs. Okay, the role of bacteria is mainly parasitic. Some of them will also be mutualistic symbiotic relationships. Fine then. Morphology of bacteria. Bacteria range their size. Like size ranges from length of bacteria can be 2 to 5 micrometers in diameters. Breadth of bacteria is 0.5 to 1 micrometers in diameter. If you look into the shape of bacteria, it is determined by the rigid cell wall it has. And the cell wall of bacteria will have what composition? Peptidoglycan in its composition. Bacterial cell wall is having peptidoglycan in its composition. Shapes of bacteria can be cocci, where bacteria will be spherical, bacilli, which is rod shaped bacteria, spirillum, bacteria will be spherical shaped, vibrios, where bacteria is comma shaped. These bacteria are pleomorphic, shapes varies with environment and nutrients. For example, bacteria is Acetobacter. Now here we see some more bacterial arrangements in cocci forms. Monococcus, where it has single cell. Diplococcus, pair of cells. Yeah, Diplococcus, the bacteria will be in pairs. Tetracocci, it is a combination of 4 cells together. Cocci cells are known as tetracocci. Okay, and then Streptococcus, the cells are in the form of a linear chain. Staphylococcus, irregular cluster-like bunches. Irregular cluster-like bunches of cells are addressed as Staphylococcus cells. Next, Sarcinae, these are cubes of 8 cells. Okay, Sarcinae, these are cubes of 8 cells. Okay, fine, right? So we have different types of cocci forms: monococcus, diplococcus, tetracoccus, streptococcus, staphylococci and Sarcinae. Some of the spiral forms are also present, which are flexible, and they are known as spirochetes. Okay, they are known as spirochetes. Others will occur as filamentous chains. Others will occur as filamentous chains. For example, Bigiotova. Next. So these are different cocci arrangements. Just now we have gone through, right? Yeah. Now Bacillus arrangement. Bacilli forms are rod shaped bacteria, right ma? Bacillus forms include rod shaped bacteria. Fine, right? Rod shaped bacteria, where it can be single bacillus, diplobacillus, pair of bacilli, streptobacilli, which is a chain of bacillus. Okay, you have to be clear with different forms of cocci and different forms of bacilli. Now you see bacterial cell structure. Cell structure of bacteria, where bacteria is outermost covered by a cell envelope. Cell envelope will have outermost layer known as glycocalyx. This glycocalyx can be either in the form of capsule or in the form of slime layer. Okay, if it is capsule, it has very thick and tight strands of glucosamine. Okay, it has very thick and tight strands of glucose. If it is slime layer, the strands are loosely arranged. Okay, if outermost layer is capsule in a bacteria, that will help to hide itself from host immune system. If the outermost layer is slime layer, it will help the bacteria from the loss of water and minerals in the host immune system. Next is cell wall. Next layer to glycocalyx is the first layer, followed by glycocalyx. It has cell wall. Cell wall is made up of peptidoglycan. Cell wall is made up of peptidoglycan. And then you have plasma membrane. Plasma membrane will have invaginations, either lamellar or tubular, or vesicle-like structures from plasma membrane are faced towards the cytoplasm. These invaginations are known as mesosomes. Mesosomes has broad range of functions. It will increase the surface area of the plasma membrane. thereby promotes respiration, releases respiratory enzyme, and this conducts the activity of respiration in the prokaryotic cell. That is the reason mesosomes of a prokaryotic cell are addressed as mitochondria of eukaryotic cell. Like mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Fine, right? I hope you are clear with 3 layers of cell envelope. Now, if we look into the cytoplasm, we have 2 types of genetic material. One is genophore, that is chromosomal DNA, and the other is extra chromosomal DNA, that is plasmid. Okay, this is genophore chromosomal DNA and this is extra chromosomal DNA, that is plasmid. Both of them will have double stranded circular DNA only. Both of them has double stranded circular DNA. And inside cytoplasm, there are no membrane bound organelles in the prokaryotic cell or bacterial cell. Okay, there are no membrane bound organelles. Fine, right? No membrane bound organelles. Now, inside cytoplasm you have ribosomes. Ribosomes are of 70S ribosomes. They are 70S ribosomes, wherein larger subunit is 50S, smaller subunit is 30S. Okay, this will be the ribosomes. Where are the ribosomes present in a bacterial cell? They are present both inside the cytoplasm and adjoining to the cell membrane. Now, look at the motile and non-motile structures of bacteria. The only motile structure of bacteria is flagella. Flagella are also of many types. It can be single or multiple flagella. We will learn types of flagella arrangement over bacteria. How these are arranged? Flagella will have 3 main components in it. One is basal body, hook and filament. Basal body, hook and filament. This flagella will have what microtubule arrangement? The microtubular arrangement is 9+0 microtubular arrangement. Fine, right? This is about motile structures over bacteria. Non-motile structures include pili or fimbriae. Non-motile structures include pili or fimbriae, which are also 9+0 arrangement. They will help the attachment of bacteria towards substratum. Pili will also have an additional function, that is to promote the sexual recombination. Okay. Now you look into the number of flagella it has. Monotrichous, one polar flagella. Amphitrichous, one flagellum at each end. Lophotrichous, 2 or more flagella at one pole. Peritrichous, flagella distributed all over the cell, that is peritrichous. Clear, right? Monotrichous is one polar flagella. Amphitrichous, one flagellum at each end of the cell. Lophotrichous, 2 or more flagella from one pole. Peritrichous, flagella if it is present all over the surface of the bacteria, that is known as peritrichous. Clear till here, ma? Any doubt anywhere? Yeah. Now, plasmid DNA. Apart from the genophore, that is main chromosomal DNA, extra chromosomal DNA which is present is plasmid DNA. It has many roles to perform. This is extra chromosomal DNA. Plasmid DNA is also addressed as non-genetic DNA. Non-genomic DNA also it is addressed as. Okay. The main characteristic features of plasmid DNA includes, it gives specific phenotypic character to the bacteria. Plasmid DNA gives specific phenotypic character to the bacteria. It aids in resisting the antibiotics. Okay, it provides resistance to antibiotics. And most important is plasmid DNA, if at all it has F factor, it also aids in sexual recombination, that is conjugation. This is about plasmid DNA. Now, nutrition in bacteria. Bacterial nutrition is mainly determined by source of carbon and source of energy. Okay, so these are photoautotrophs. They use light energy to make food. Okay, these are photoautotrophs. They use light energy to make food, and carbon from carbon dioxide. Okay, here in photoautotrophs, from where carbon and light energy is being taken from sunlight, and carbon is from carbon dioxide. Clear, right? Fine, right? So here photoautotrophs, they use light energy as a source of energy and carbon from carbon dioxide. For example, here you see what are the examples of photoautotrophs? Chromatium and chlorobium. These are the examples of photoautotrophs. Chemoautotrophs, they use inorganic chemicals. They use inorganic chemicals and no sunlight or organics. They derive energy by oxidation of inorganic substances. Here the energy is from sunlight, but here it is from oxidation of inorganic substances. Carbon source is the same, that is from carbon dioxide. Okay. Examples if you observe: Nitrosomonas, Nitrobacter, Bigiotova and Methanogens. These are the examples of chemoautotrophs. Yeah. Photoheterotrophs, the name itself is saying, right? Photo means the light energy will be sunlight. Heterotrophs means they are not oxidizing by themselves. Carbon is from other source. Carbon is from organic compounds here. They are taking up carbon from organic compounds. Example is Rhodospirillum and Rhodopseudomonas. Chemoheterotrophs, they use organic molecules. Chemoheterotrophs will use organic molecules for both carbon and energy. That is the reason they are chemoheterotrophs. Nowhere the energy is from sunlight. Energy from respiration or fermentation is taken. When ATP is produced during respiration or fermentation, that ATP is utilized as energy source. Fine, right? So this is about different types of nutrition. Most important point to be addressed: the drastic diversity, high amount of diversity in metabolism is observed in bacteria, both nutritional diversity and respiration diversity. Okay, both anabolic and catabolic diversity is observed in bacteria. Here we also have subtypes in chemoheterotrophs. Subtype saprophytes. Characteristic is it is used to, it feeds on dead organic matter, that is detritus. Example is Bacillus species. Parasites, they derive nutrients from living host cells or tissues. Example Xanthomonas or Salmonella. Clear till here? Any doubt anywhere? And then reproduction. The most common mode of reproduction in bacteria is binary fission, which is most common. As most of the bacteria or the prokaryotes are unicellular, the basic mode of division is binary fission, which is also addressed as amitosis. As there is no involvement of spindle apparatus for the cell to divide, it will divide eventually. Like karyokinesis will happen followed by cytokinesis without the involvement of any spindle fibers. Now sexual reproduction. Rather than addressing it as sexual reproduction, do address it as sexual recombination, wherein first mode of sexual recombination is conjugation. Here you look into it. In conjugation, what happens is, those bacteria in whose plasmid DNA F factor is present, that is addressed as F plus strain. And in the bacteria which doesn't have F plasmid are known as F minus strain. Fine, right? Now from F plus strain, small amount of F factor will be transferred to F minus strain via pilus or pili, which is also known as sex pilus or sex pili. Okay, wherein through this sex pili, the F factor from F plus strain will be transmitted to F minus strain. Thereby, in the products if you see, you will see both the daughter cells with F factor, F plus strains. Clear, right? So, this is about conjugation. Once look into the theory also, direct transfer of DNA will take place between donor cell and to the recipient cell. It is discovered by Lederberg and Tatum in the year 1946. Okay, this is discovered by Lederberg and Tatum in the year 1946 in Escherichia coli. Main mechanism here is this requires sex pilus or conjugation tube, where F plus has F plasmid, F minus cells doesn't have F plasmid. Pilus contracts, drawing out the cells together. Okay, the sex pili will draw out the cells together, wherein DNA replicates from the plasmid DNA and this gets transferred to the F minus strain. Okay, donor retains a copy. The process of replication over here you can see is conservative method. Next is transformation, which is uptake of naked DNA fragments from the environment by bacterial cell. This is given by Frederick Griffith in the year 1928 on the bacterium Streptococcus pneumoniae. Okay, wherein the bacteria will take up the DNA. The genetic transfer is from the environment in which the bacteria is. It is not cell to cell transfer. Here DNA is absorbed by the recipient from the surrounding environments. This results in genetic alteration and the recipient will acquire new traits as such. Next is transduction. Transduction is due to viral infection on bacteria. The transfer of genetic information from virus to bacteria is known as transduction. Here the transfer of DNA from one bacteria to another via bacteriophage. What is bacteriophage? The virus which is infecting bacteria. Discovery, this is discovered by Lederberg and Zinder. Okay, in the year 1951 in Salmonella typhimurium. Okay, this is discovered in Salmonella typhimurium. The importance of bacteria in human industrial applications, where lactic acid bacteria is used in curd production, vinegar production which is done by Acetobacter aceti, alcohol production by different modes of bacteria, Zymomonas, Clostridium and Saccharomyces. These are the bacteria which is used in industrial applications. Antibiotics, Streptomyces griseus will produce streptomycin, Bacillus subtilis, bacitracin. Penicillium will give penicillin. Enzymes produced for industrial use, steroid transformations. This is performed by Corynebacterium. Okay, and then in agriculture for nitrogen fixation, gram-negative bacteria, that is Rhizobium, is used, which is obligate symbiont for leguminous plants. Free-living bacteria which fixes nitrogen includes Acetobacter and Clostridium. Nitrifying bacteria, Nitrosomonas and Nitrobacter. Nitrosomonas which converts ammonia to nitrites, Nitrobacter from nitrites to nitrates. Those bacteria which are used as biopesticides, Bacillus thuringiensis, and they are also used in decomposers which will help to recycle the nutrients in the ecosystem. Here is the list of useful and harmful bacteria. Go through that carefully. If you want, you can take a screenshot of it also. For dairy industry, lactobacillus, whereas clostridium is used in food poisoning. Nitrogen fixation, rhizobium, acetobacter. Here harmful bacterium is Pseudomonas denitrificans. Antibiotic production is Streptomyces griseus. Biopesticides, Bacillus thuringiensis, and here harmful bacteria, that is Erwinia amylovora. Fermentation, Clostridium is useful, and Salmonella typhi will cause typhoid fever. Yeah, these are the bacteria which we have exercised just now. Just go through the harmful bacteria also. Yeah, you can take a screenshot of it now, it is useful. Yeah, so with this the chapter bacteria is done. What all we have learned, starting from the bacterial distribution, bacterial introduction, discoveries of bacteria, followed by different shapes of bacteria, sizes of bacteria, followed by prokaryotic cell, and then nutritional diversity in bacteria, followed by reproduction, both asexual reproduction, binary fission, and sexual reproduction. We have looked into conjugation, transduction, transformation. That's all, right? Yeah, that's all for today's session. In the next session, we'll look into viruses, post which we'll solve complete EAPCET PYQs on microbiology. Clear, right? That's all for today's session, and thank you. Yeah, I'll address this query in the comment section. In higher classes, if you take 11th and 12th standards, many concepts from your high school will be connected. Like if you see cell biology or reproduction or heredity, these are the concepts which are in connection with your higher standards. The more you increase in the concept, more memorizing points also are there to remember. Biology is the subject which I feel where you have to remember more. Okay, how you can memorize the things more? When you revise it repeatedly. Okay, conceptual understanding is very important for applied biology concepts, whereas core botany or core zoology concepts will require more of revision so that you can memorize the points in a clear way. Okay, so this is what I can address from the query which is given in the comment.